Well, thank you very much. I very much appreciate the invitation, to participate in this outstanding Congress, and also sort of the challenge of trying to look at the year in review in lupus. What I will do is present to you a story of the hundreds of papers that have been published in the last year or so on lupus. The good side of this is I can tell you a story because what is emerging is a very interesting and really intellectually coherent view of lupus. Now, the downside is it a true story or not? And that is something we will begin to discuss. So this is the story I'll tell you, and it's based on sort of the current model of lupus, which posits that autoimmunity in this disease results from an increase in the amount of extracellular nuclear antigens, and that these antigens can form complexes. And once the complexes are formed, sort of new immunological activities are created because of the now structure that's developed, and that can drive the production of type 1 interferon and also other cytokines, I should add, by a particular cell population called plasmacytoid dendritic cells, which are highly specialized for this type of response in innate immunity. And that the molecules that are involved in this response are both toll-like and non-toll-like receptors that respond to nucleic acids, and they are key to the activation of the PDCs. You may ask, why are there sensors for nucleic acids? And the answer usually given is intracellular infection, whether it's bacterial or viruses. So there have to be signaling systems for cells that are currently infected, and that these are on the insides of the cells, and they respond to nucleic acids. But this system can be subverted if DNA or RNA has access to the internal sensing systems. So studies on mechanisms to develop new therapy thus focus on the process that lead to a number of steps in this, and that is nuclear antigen release, formation of complexes, and then now the downstream signaling events. So the first area I want to talk about, which you heard about the other day in a very beautiful talk from Mariana Kaplan, is the story of nets. Nets are neutrophil extracellular traps discovered about a decade ago, and they were really discovered originally in the context of infection as sort of a novel strategy by which neutrophils can defend against foreign organisms, and they use a physical modality, which is to the development of a very extended structure, which is a mesh-like or net-like structure, during a process of cell death or cell activation, which is called netosis, because it leads to the elaboration of these structures. And in this process, antibacterial proteins that are usually in the cytoplasm mix with the DNA from chromatin to form these structures. And as previously shown by other investigators, including Mariana, is that nets can activate PDCs to produce interferon. They have other actions, again, from her work, to damage endothelial cells. And one could then begin to imagine that lupus is an imbalance of net generation and net clearance. Significance here is, again, the production of extracellular nuclear material, antigens that can form immune complex, and also a direct mechanism to damage endothelium, which could begin to explain perhaps the cardiovascular issues in terms of lupus. This is a picture by other investigators. It's been colored to illustrate, and what you see is this mesh-like structure in yellow, and what is in orange are bacteria, and these have been physically entrapped in the nets, and because of the antibacterial proteins, the bacteria are killed. So the first paper I'll talk about is a very interesting study from Mariana Kaplan and her group, looking at extracellular traps associated activation, now of a different system. This is called the NLP, the NALP3 inflammasome, is enhanced in lupus macrophages, not PDCs. Now, the inflammasome is another sensing system in the body, in the innate immune system. There are toll-like receptors that lead to the elaboration of TNF, and then there's the inflammasome, which can be triggered by really a host of different stimuli, and it leads to IL-1. So the significance here is a different triggering system, but also a different downstream product, and that product is IL-1. And as shown here, this is looking at the nets, and what this shows is that the nets have proteins that would be expected, including LL37, which is a defensin. It's a cellular protein that can bind DNA, but also has antibacterial antibodies. myeloperoxidate. These are the very pretty structures that can get formed, which is these meshes or strands of DNA which can be identified by fluorescence. These can be produced by a number of cell types, again, mostly neutrophils, in particular a population called low-density granulocytes, which are increased in lupus. This is really the important study right here in panel C, and what is done here is to take macrophages, activate them in LPS so that they can respond and via the inflammasome, and then what is done is to stimulate them with the NET, shown here in dark, or ATP, which stimulates the inflammasome by a purinergic receptor. The important point here is that the NETs drive the production of both IL-1 and IL-18, which are not cytokines we usually think of as being involved in lupus. This panel just shows activation of caspase. Important point here is that in this instance is this protein called LL37, which is responsible for driving the activation of the inflammasome. So this identifies still another molecule that could be playing a role in lupus. Another feature of this system is that the macrophages from patients with lupus are more responsive to the NETs than ordinary macrophages. Again, using LL37 as a stimulus, black is shown as lupus macrophages, unfilled boxes are controls, and what you can see is that the macrophages from the patients with lupus are more responsive. Again, driving IL-1 production and IL-18, and this is the mechanism. Neutrophils get activated, produce the structures, including LL37, stimulate receptors, and then drive these cytokines, and that's IL-1 beta and IL-18. These are cytokines we usually don't think of involved in lupus. Important thing here is we have approaches now if we wanted to eliminate them. Another aspect of the NET story is what happens in patients with lupus. Normally or ordinarily, we think that there should be degradation of the NETs, mostly in terms of their nucleic acid constituents. And what this study, which is a follow-up of a previous study that looked at it, showed that the traps that are formed are not degraded in lupus, and that furthermore, these NETs can activate complement. In this study, it shows groups of patients with lupus that you can, when they are in remission, they degrade, almost like patients who are normal. When patients flare, they lose their degradation capacity. This just, again, compares net degradation with remission and with a flare. If you add DNase to this, there is increased net degradation, not quite back to normal, and that one can look at this process, again, as a biomarker for disease activity. This capacity for degrading nets is associated with the ability to generate type 1 interferon, and so that those individuals who do not degrade nets well have high interferon, and this is associated with serological activity. So those individuals with high DNA, histones or C1q, are impaired in their ability to bind the net, but the nets also bind DNA, can form immune complex, and they can activate complement. So the significance of this is that the number of important significance. One, neutrophils can be a source of the DNA. We usually think of apoptotic cells. This is another cell population that can do it. The inflammasome, a new player in terms of lupus. Again, we have not been thinking of that system. IL-1 and IL-18 as potential mediators and therefore targets. Further elaboration and demonstration of abnormalities in now both PDCs and macrophages, and a removal of impaired clearance of net and generating extracellular material, either to drive inflammation or perturb the endothelium. That story is a very nice story, and again, there are always caveats, and this is a study from Mark Schlomchik that was published in Science Translational Medicine, which tried to get at the issue of the role of nets by using a knockout. This is the NADPH oxidase system, which is involved in net formation. So in the absence of NADH oxidase, one would expect there would be impaired net formation, impaired generation of extracellular nucleic acid, and therefore improvement in lupus. And using an animal model, what he showed was the exact opposite. And so that the knockout mice that couldn't putatively generate nets had intact antibody responses, whether to SM or RNA. importantly, they had increased disease activity. So this is not what you would have expected, and this was also observed even in the heterozygote. This is an X-linked gene, so female heterozygotes really also had disease. So while one would have predicted that a system that's involved in net generation, inhibition would attenuate disease, the opposite's case. And so one could always say that NADPH oxidase is doing something else, or the alternative is that maybe nets are not as involved as we think. And this is a very interesting area, but also unresolved. Now, as I mentioned, there's a lot of interest in the role of the plasmacytoid dendritic cells and what they are able to do. And what we're now going to see is a number of genetic models that begin to explore this paradigm essentially by knocking out certain genes that would be involved in plasmacytoid dendritic cells. I should say, in all the animal studies, these studies have been done in a number of different mouse models, NZB, MRL, B6LPR, BXSB. Reason's not clear why people have used so many different models to look at the same thing, but they have. I'm just going to call them mouse models of lupus. There may be nuances here, but for simplicity, we'll do it that way. So there are two gene systems. One is IRF8. It's an interferon responsive gene. Important here is in the absence of that gene, plasmacytoid dendritic cells do not form. And the other is this SLC15A4, which is involved in a transporter that allows toll-like signaling to occur. So while the PDCs can form, they don't work. So what was done here first in an NZB model is to cross these mice so that they lack IRF8. They don't make plasmacytoid dendritic cells. And the outcome of that is an impairment in the production of anti-chromatin antibodies as a model of lupus, both IgM, IgG. There's less immune complex formation. There's less glomerulonephritis. And in another marker system, less red blood cells. So in this system, an absence of PDCs is involved. You may ask why red blood cells should be affected. Red blood cells don't have DNA in them, although they do have some RNA, and that may suggest where the origin is really in the bone marrow as opposed to the periphery. Anti-nuclear antibody production is attenuated, as is immune complex deposition. So eliminating PDCs has a profound impact in this model. This is a sort of another model, again, using a slightly different strain. This is BLAC6LPR. This is now to put in a knockout for this gene. And again, while the PDCs are there, they do not work because toll-like signaling does not go forward. And the bottom line is, again, illustrated here. That is a diminution of the anti-chromatin response, less anti-nuclear antibody production, and improved survival. So what this supports is the idea that PDCs are very importantly involved in terms of their ability to probably produce cytokines or other roles. Again, a popular concept that the downstream effects of this stimulation in terms of type 1 interferon would drive events in lupus. This is a system that explores this in terms of using an antibody to the interferon receptor, which has obvious analogies between treatments that are currently ongoing. And again, in this mouse model, the treatment with the antibody, which covers the cells, which affects the number of cells, as illustrated here, again, leads to less autoantibody production, improvement in clinical events, and improved survival. An important feature of this story, however, is timing. So when therapy with the anti-interferon antibody was started early, it was effective. When it was started late in the course of disease, there was much less benefit. So I think this is, something to be borne in mind as people do clinical trials, is when we are treating. And the failure of a response in people with long-standing, well-established disease really may not indicate that interferon is not involved. This is a very interesting study from Keith Elkond's group, which is a double transgenic system. Now, as I said, the sensors for nucleic acids are on the inside of cells, so the tolls occupy space in the endosome, and that's presumably because they respond to internal DNA, and that DNA or RNA usually gets inside by being transported in, and the usual transporter turns out to be antibody. So what Keith did and his colleagues was to engineer a mouse that had increased expression of RNase in the extracellular form. So this is an RNase transgenic mouse under the control of an albumin promoter, so it's going to be secreted. So the RNase in this case is on the outside, it's in the blood. Nevertheless, as illustrated here, in a double transgenic, TLR7 transgenic mice develop an autoimmune disease. They also develop hepatitis, probably from hepatic inflammation from too much toll stimulation. And the double transgenic mice have reduced disease manifestations in terms of splenomegaly, and again, a number of cellular changes. But the, again, important point here is the double transgenic mice are protected. So extracellular RNase protects against stimulation, and that is seen with reduction of autoantibody levels as well. Now, in the past, there was effort to treat lupus with DNase extracellular, which was not considered successful. Whether RNase is better than DNase would be speculative, but then again, it raises questions, but what's the nature of this extracellular nucleic acid that really is now sensitive to digestion and susceptible? Now, the story that I was telling you really has been much based about the role of the tolls, particularly those that respond to DNA and RNA, and that's toll 7 for single-stranded, toll 9 for double-stranded, for DNA. Toll 3 is also involved. We give it less attention. That responds to double-stranded RNA. Rainer Voll, working in Freiburg originally, made an observation a number of years ago about the nature of the immune complexes in lupus, and his model posited that the immune complexes in lupus are built of nucleosomes, but the nucleosomes also have a companion protein called high mobility group 1 protein, HMGB1. HMGB1 is a prototype of what is called an alarmin. It's, again, a nuclear protein with immunological activity. Important aspect of HMGB1 is it can stimulate a number of tolls themselves, and that is toll2, TEL4, and it also can stimulate RAGE. And he showed that in an immunization model, toll2 is involved. Again, somewhat a surprise because the antigens that we're looking at would be expected to be toll9 or toll7 dependent. And he created, again, a mouse that lacked toll2, and this was the pristane-induced model of autoimmunity, which has been pioneered by Wes Reeves, involving mineral oil, and these mice eventually make anti-nuclear antibodies. They get formation of lipogranulomas, which drive this response. And the important result here is that a toll2 was very much involved in that. So while we think of the toll-like receptors being involved in lupus as being those that respond to nucleic acids, again, 7 and 9, there's a role of other tolls, and for those interested in using anti-toll-like strategies for therapy, this really widens the opportunities in terms of other tolls. Toll2 usually responds to bacterial protein, although again, like many tolls, it responds to intracellular material. Again, this is just a further demonstration of what happens when toll2 is not there. Cell numbers go down, plasma cells go down, bone marrow plasma cells go down. So it's highly effective, really, in blunting this, this model of autoimmunity. Now, again, what all this is bringing up is the importance of serology, and that to get these responses, autoantibodies are key because autoantibodies form an immune complex which either can deposit in the kidney or activate cells by sort of translocating DNA or RNA into the cell to access these receptors. And this is using a novel technology to get very, very sensitive assays for anti-nuclear antibody. And what this group showed is that in lupus, in fact, autoantibodies occur in clusters, something we recognized before. SM and RNP occur together a lot. Rho and La occur together a lot. However, what this study showed using this enhanced technology is that they are frequently not produced together. So those who are anti-SM RNP positive are one group. They tend not to produce as much anti-Rho or anti-LA. On the other hand, there are individuals who make a lot of anti-Rho and anti-LA, and their production of SM RNP is much lower. Now, this addresses an interesting question. What is the specificity from? And the answer is unknown, but this just indicates that the RNA protein complexes may have different biology. There is some limited clinical association here in terms of differences in serositis. But the major point here is even though lupus has lots of autoantibodies, it is regulated and there is specificity. Now, this is a study from Tim Newall, who's really done outstanding work in terms of trying to dissect the genetics of lupus. Now, one of the associations that has occurred is so-called IRF5, which is, again, an interferon-responsive gene. There is evidence that this plays an important role, and again, in terms of the story that I've been telling you, it would make sense that something in the interferon system would somehow promote disease. And what Tim's study did was really add another dimension to the story and said that the effect of this particular system really becomes apparent when you look at the autoantibodies that are present. So if you look at different risk alleles in these populations, in the absence of serology, in this case anti-DNA, you do not observe much in terms of differences measuring serum interferon activity. On the other hand, when you include serology, such as anti-DNA, you begin to see now the effects of these at-risk haplotypes in increasing the amount of interferon. That is also true of anti-Rho. This is African-American population, but this particular risk comes of people from European ancestry. And what he created as a model, a very interesting model that says the autoantibodies are involved, they form immune complexes which drive interferon, and depending what the risk allele is, that will lead to increased interferon, although it is most certainly possible that this is a feedback or feed-forward system, and that high interferon then leads to greater generation of autoantibodies. Important point here, it's the combination of genetics plus serology that allows you to see this increased interferon. Now, this is a story that's near to my heart because I've been involved in, and that is another way of looking at immune complexes in lupus, which is another structure that forms when cells get activated or cells die, which are called microparticles. These are small membrane-bound structures. The simplest way to think of them, these are the apoptotic blebs that Tony Rosen has talked that now have left the cell. So during cell death, there's a lot of translocation and movement of nuclear molecules. They go into the blebs, but as this process goes on, the blebs pop off, and they're in the circulation. And just like nets, particles contain nucleic acids. They're immunologically very active, and they also have effects on endothelium. So this is an important source of extracellular nuclear material, and again, by a group from Denmark, what they were able to show is that these microparticles, in fact, are a source of immune complexes. Particles can be measured by flow cytometry. They're small. People who do flow cytometry usually ignore them, but they're always there. But one could look in the blood of patients with lupus, and what is observed is increases amount of these particles that have IgG on them, IgM, and C1q. So they're bona fide immune complexes, and they are in the circulation. Just an important technical issue here is you see them if you use plasma. If you use serum, it's much higher. Most people use serum. So this is just to extend methodologically the importance of plasma. If you again look at these, while the total number of particles are not increased necessarily in patients with lupus, the big difference is in terms of the number of immune complex-like particles. And again, the interesting area here is going to be the relative contribution of nets versus particles as a source of nucleic acid and what particles do. This is a study from my own group looking at mice, and mice are like men in this, and that the autoimmune mice also produce autoantibodies that can bind to particles, and in their blood they have circulating immune complexes, and this is just shown by flow cytometry. And in this study we saw it much more apparently with antibodies from, or plasma, I'm sorry, from the MRL strain than we did with the NZB mice. Having said that, MRL make a lot more anti-DNA. So both humans as well as mice have particles that can serve as immune complexes. We do not have good models for immune complex behavior of particles as opposed to regular antigens. So an interesting topic for the future is to understand particle biology and how it may impact on responses. Now, there's been a lot of interest in other strategies to block production of lupus, including proteasome inhibitors. A number of investigators, particularly Jennifer Analik and John Looney, have been very involved in this. And they did, I should also say, Rainer Voll originally, who did studies using bortezomib, which is approved for the treatment of plasma cell diseases. It impairs the function of the proteasome, which is the degradation system in the cell. In the absence of degradation, the unfolded protein response gets induced and cells die. So it's been highly effective as a treatment of plasma cell dyscrasias and myelomas, and by extension, since plasma cells are involved in lupus, originally Rainer Voll and Joachim Kolden explored this as a therapy and showed that treatment of NZBW mice with this inhibitor, in fact, blocked disease. This study is to look at other proteasome inhibitors beyond bortezomib. This is called fizimab, and this is a new compound which just has a name. The difference is bortezomib was associated with neuropathy. This one had less neuropathy, and this inhibitor also was more specific for the so-called immunoproteasome, which is involved in immune cells. The important point here is inhibiting this system either non-specifically or more specifically blocks disease in MRL, NZBW mice, leads to difference of anti-DNA production, but the interesting result here is that there is an effect on the generation of interferon. So the effect is not only on plasma cells potentially or B cells, it's also in the interferon-producing cells. So finally, again, something that I think we've been talking about a great deal at this meeting is the effects of hydroxychloroquine. As you heard just from Jill's talk, hydroxychloroquine is very much involved in signaling by toll-like receptors. There's an acidification step involved. Hydroxychloroquine and chloroquine are bases inside the cell. They block toll-like stimulation. And this is just to look at patients with lupus treated with hydroxychloroquine ex vivo. Their cells don't respond well to stimulation of either toll-9 or via toll-7. So although we are talking about strategies to block this, already we've been doing it probably unbeknownst to us, probably rather effectively. And as you've heard, there have been so many good effects of Plaquenil. And again, it would be speculative as this is what the action. So to really summarize here, my take on the year in review, emergence of the neutrophil. Not a cell we've talked about a lot. It's an important player probably both in cytokine production and nets. Still much information on the role of another cell type, the PDCs. Involvement of more than the tolls that we've been thinking about, not just toll 7 and 9, but also toll 2, probably 4. Identification of new types of immune complexes, and also identification of new targets of therapy. Thank you very much.
The very last slide that you showed, I thought it was intriguing because sometimes you don't see everything at once. It looked like on the bottom panel that actually hydroxychloroquine was not significantly associated with the interferon signature, but rather the TNF-alpha. Did I see that wrong? Because I think that is a point that people don't really emphasize. We're very, interferon-centric.
Yeah.
Yet, there's this whole macrophage world where, in fact, it's not interferon alpha that's generated, but it's TNF.
From what I've heard at this meeting, although we think of the interferon signature as indicative of type 1 interferon, that may not be fully correct. And that other interferons, including gamma, can give a very similar picture. So although you may block this type of pathway, you're not blocking the production of gamma as far as we know. And so there may be persistence of interferon signatures even in people with Plaquenil because there are other pathways to get there. So again, this was a story about the role of type 1 as opposed to type 2 interferons. And since other interferons can generate their signatures, again, you may be able to have blocked this particular aspect, but not the whole story. But this does say a relatively benign drug really has powerful immunological effects, and although we don't really think of it that way, it's really blocking a fundamental pathway quite effectively.
A place where there might be better development is if we could balance an elimination of ocular toxicity with better, because I think most of us confine ourselves to 6.5 milligrams per kilogram of hydroxychloroquine, right? And the kids are getting obviously very low doses, the Asian, and maybe we are really underselling ourselves, and I mean, that's just a balance we kind of have to live with, right?
You know, Plaquenil was applied to lupus empirically without dose finding, with the restriction on the ocular toxicity. There have been limited efforts, believe it or not, to figure out what Plaquenil really does, and could you create a Plaquenil without ocular toxicity. Some of that, there are other actions. Plaquenil can also inhibit an enzyme called quinone reductase 2. And there's another enzyme that inhibits, it's an aldehyde dehydrogenase in the eye. So one idea has been the ocular toxicity is really an off-target effect in the eye, and that it would certainly be possible to generate another derivative that just was, you know, either base or whatever that doesn't have the ocular toxicity. Now, Peter Leonard at Iowa actually has many Plaquenils-like drugs that have really not gone on to development. Now, it's quite possible, you know, if you could get around the ocular toxicity, then, in fact, you could give even higher doses and get more effect. But it's just a matter of trying to develop new compounds that separate out the ocular toxicity from either the enzyme inhibition effect or serving as a base to get into the toll-like endosomal system. It just hasn't been done. You know, the answer is they're all drugs. It's actually interesting about anti-malaria. It's not even clear why it works in malaria, because apparently the drugs don't go after the organism. It's actually making the host incompatible with malaria. And that's why resistance to antimalarials takes place differently than a conventional antibiotic, because it's more at the host. But again, there's been very limited development of other compounds in this pathway. But since we know small molecules, you know, it's a small molecule, well absorbed, well tolerated, to me it would seem very worthwhile to see if you could push the dose, you know, to get more efficacy. But, okay.